So fruit flies are very behaviorally diverse. This is especially true for their courtship patterns. So males perform a variety of different courtship displays in front of females when attempting to mate. And closely related species have very different patterns of display activity. So here you're looking at a group of different Drosophila species in the melanogaster group. And hopefully you can see that the males perform a variety of display characteristics in front of the females. And so courtship behavior in males is a rapidly evolving trait in fruit flies. So these differences often involve variation in wing activity. So some of you may know that Drosophila have a variety of different songs that they perform. And these songs evolve rapidly in different males, but they also involve variation in different wing activity levels. So what you're looking at here is a male Drosophila elegans fly performing display activity in front of the female. And hopefully you can see that these flies have pigmented wing spots that become visible whenever they're dancing in front of the females. So next I'll show you a video of Drosophila elegans as it performs this wing display activity in front of the females during courtship. You can see as the male attempts to mate, he'll dance sort of like a matador in front of the female before attempting to copulate. But what's really interesting is a closely related relative, its closest relative, Drosophila gargancola, does not perform any wing display activity during development. So here's a video illustrating their behavioral activity. So all these males do is chase the female. They do perform some song, and then they'll attempt to mount her as the courtship proceeds. So these guys have lost all aspects of wing display activity, and you may not be able to see, but these flies also have lost their wing spots. And so what's really interesting about this system, this group, is the presence of display activity and the presence of wing spots, or the absence, has been gained and lost multiple times across this lineage. And what you can see in this phylogeny is that those males that possess the wing spots also possess wing display activity. When the wing spots are lost, the display activity is also lost. So the question that I'd like to be able to answer is how complex traits like these, like those involving wing display activity, evolve between species. And answering this question really requires a way to reliably quantify behavioral differences that are occurring. So what I've been using so far in my research is a newly developed tracker. So this is called Caltech Fly Tracker. It's being developed at the California Institute of Technology. It's not yet published, but it will be later this year. And what's cool about this tracker is it can keep track of not only the body movements of the flies, which we've been able to do for a number of years now, but it can also, in a reliable way, keep track of the wing movements and the leg movements in freely moving animals. So that's what's novel about this tracking technology. So Arun Oljolfsdotter at the California Institute of Technology really developed this software, and it's freely available for download. If you guys are interested in doing this type of tracking work in your own research, at this website below. So I applied this tracker to study courtship differences between these species. So on the left is what I'm calling the spotted species, like you saw earlier, Drosophila elegans, and on the right is a male of the unspotted species, Drosophila gargancola. So the way this tracker works is, like I said, it keeps track of the body and the wings in freely moving animals, and it also keeps track of a number of individual features as the fly freely moves about. The features that I'd really like you to pay attention here are variation in wing display levels. So it'll keep track of both the minimum and the maximum wing angles, which are highlighted in 3 and 4 respectively in this figure. And what I've done and what you'll see on the next figure is quantify 2 wing angle measurement differences. So that's really part 3 and part 4 added together. So on the y-axis in this figure, what you're seeing is variation in that 2 wing angle activity as the fly freely moves about in the arena. And this is one individual as it courts a single female fly. And so this is in 20 minutes illustrating the wing display activity levels across fly as he's courting a single male, And in Drosophila, you can see that these levels are clearly lower, indicating that the tracker is doing actually a fairly robust job of keeping track of the wing display trait as it occurs during courtship. So you can highlight and annotate some of these behaviors. So mid-level peaks here correspond, interestingly, to one wing extension activity or unilateral wing extension activity, whereas the taller peaks that climb to above 200 degrees, they actually hyperextend their wings past 180 degrees, correspond fairly well to the two-wing extension. These lower peaks and broader peaks often correspond to wing grooming activity. As you can see, the Gongocola species doesn't really have any of the displays. The lower peaks correspond to attempts in copulation, so it's highly sensitive, and then those other peaks also correspond to wing grooming behavior. So here I'm just trying to illustrate a population level of the differences. So these are just individual flies that have been compiled on top of each other, just to give you an idea of the variation in the display trait. But for the most part, the tracker is accurately quantifying these display traits across several individuals, and the correlation between manual measurements is actually fairly tight. So by looking at a number of individuals, we can then calculate interesting statistics to test questions about behaviors. What you can see here is the brown bar corresponds to the average maximum wing display angle that occurs in this wild type species. So on average, these guys perform wing display activities at 197 degrees, whereas in Gongocola, they don't have the trait, so there is no average that display. What I'd like to illustrate here is these are going to be histograms, and on the y-axis is variation between a threshold of wing display activity. So it's between 100 and 220 degrees. The reason I chose these thresholds is because from all of my annotations, these are the wing display angles that actually correspond to the wing display activity in the behaving animal. So this is just a single individual to illustrate that we can get interesting time activity data from this tracker information. So for the single individual, out of 20 minutes, this guy spent about 119 seconds with his wing extended past 100 degrees. You can use that to answer interesting questions about individual variation. So I'd also like to highlight its ability to get at qualitative differences in position. So this tracker will give you XY positioning throughout the arena as the fly moves, and you can map that just to get an idea of where he's going. So my arenas are circular shaped. These guys look like they spend some more time maybe towards the center of the arena, whereas this species, interestingly, spends most of its time on the periphery. And I think what you're looking at here is a qualitative depiction of the fly spending most of its time chasing the female, whereas in the other species, in elegans, this guy is dancing, chasing behavior occurs, but it's less frequent, and so he spends more time in the middle. These are just multiple individuals mapped on top of each other to illustrate that. And so you can see there's a lot more density in the center of the arena for elegans, whereas for Gongocola, the spotless species, they really spend their time on the periphery. Okay, so what I'm really curious about is how these differences in wing activity and the locomotion patterns have evolved between species. And what's really cool about this system is you can actually make hybrids and do backcross experiments to study the genetic basis of behavior. So what I'm going to show you in the rest of the talk is really just variation between hybrids. So what you can do is take a male Gongocola fly and cross into an elegant female. And that can produce an F1 hybrid, illustrated down here in the picture below, that has spots. And importantly, what I'd like you to notice is that this guy has his X chromosome from the spotted species, from the elegant species. Okay? You can do the reciprocal cross, where you can take a male elegant, cross it to a female Gongocola. You can produce a hybrid that is identical to the previous hybrid, except for the X chromosome. In this case, the X chromosome is coming from the Gongocola species. These guys, interestingly, don't have spots, suggesting the importance of the X chromosome in spot development. Okay, so I'm going to show you the same plots that I just did previously, but now we're comparing hybrids. On the left are hybrids with the X chromosome from the spotted species. On the right are hybrids with the X chromosome from Gongocola, the unspotted species. What you can hopefully see is that their display variation looks very similar to the wild-type elegant species, whereas these guys still have the display trait, but it's more variable. And so these are, again, are 6 individuals mapped on top of each other. And what I've done is calculated just average levels of wing display activity in dozens of individuals between these guys. So the red and the blue bar are statistically significantly different from each other. Importantly, so these guys on the left have about 189 degree display activity, whereas the guys on the right have about 150 degree display activity. These brown bars correspond, like I showed earlier, to the elegant wild type levels. The guys on the left are not statistically different from each other, whereas the red and the brown on the right are. So the last bit of data that I'll show you is just differences in locomotion between these guys. So these are 6 individuals on the left with the X chromosome from the spotted species, whereas on the right is the reciprocal. So you might be able to see that the guys on the left, I think, spend more time maybe towards the center, whereas the guys on the right seem to be really a mix of both. And one way you can use the tracker to quantify these differences is it can give you data on the distance from the wall of the arena. And so the y-axis here is distance to the wall. And these guys are statistically different in the amount of time that they actually spend away from the wall. So hybrids on the left spend more of their time based on this data towards the center of the arena, which is the furthest distance from the wall, whereas the guys on the right spend more of their time towards the wall, likely performing more chasing behavior like the Gummicola species. Okay, so the key point here is that behavioral differences between reciprocal F1 hybrids strongly suggest that the X chromosome has played a really important role in the evolution of these behavioral differences, because the only difference between these guys is the presence of the X chromosome from each parent. So future work aims to map specific loci that are involved in this and that are important in the species differences by genotyping and phenotyping a growing population of back cross recombinants. And what's really, I think the key thing here is that we can use this tracking technology to automate the entire process. So we can look at hundreds of videos at once instead of manually annotating them, which takes hours and hours of time, and we can get lots of rich behavioral data to be able to answer these, I think, important questions. Okay, so I'd like to acknowledge a number of labs that have helped me do this work. I obviously cannot do it alone, and I started before the timer because I spaced out a little bit, so I think I have a little bit of time for questions. Thank you for your attention.